My outline is that to emphasize that we've made tremendous progress in our ability to treat an individual patient with type 1 diabetes, but that the cure is still important, and I'll point out data as to why. I'll tell you the model that we've all worked under for the last 30 years or 35 years to develop a better understanding of diabetes pathogenesis and better treatments. A theme that I'll be discussing a lot is a lot of ideas that we think are new are actually quite old. And then I'll be puncturing a lot of standard assumptions and dogmas and showing you data that will, I hope, raise questions in your mind about a lot of things that we've assumed, which just ain't so. Some of the work I'll describe I did before I came here with Shira Pearl and John Tisdale at the NIH and a collaborator at the Lawrence Livermore Lab, Bruce Bruckholz. We get a lot of our islets from Al Powers and other sources, but I specifically like to acknowledge Al. We've got a terrific team here at UMass with Dale, Sally Kent, Mike Brem, Sam Reddick, Dave Blodgett, Renee Marr, and Jennifer Wang, and a long-standing relationship with the Jackson Laboratories and Lenny Schultz. I'll briefly describe some recent collaboration with Katerina Angeli and Yi Hao Zhang at WPI. And then we have a long-term collaborative relationship with our colleagues at Harvard, predominantly Doug Melton, but also George Daly, Jason Gallia, Stefan Kistler, Pin Yi, and Judith Agudo, which I'll mention toward the end. We have to always acknowledge appropriately the funding sponsors that allow us to do this work, including our visiting advisory committee. And then all of us that speak on this subject now acknowledge the patients, donors, and their loved ones that allow us to study human islets and human pancreatic samples. When somebody has been declared brain dead, we get their pancreas and islets for study. So the worst day of someone's life, they have to do something for the greater good, and we try to always acknowledge that. All right, so I've said type 1 diabetes remains a huge medical problem. This is a slide that you all saw a few months ago when Des Schatz delivered endocrine grand rounds. Type 1 diabetes is very prevalent, lots of new cases every year. He said shortened life expectancy, and I put an asterisk on that because of interest. We're going to dig deep into the DCCT study in a minute. But I thought this was interesting, published 4 years ago, that if you looked at the 700 people assigned to the intensive treatment arm of the DCCT and compared their mortality to the general US population, it's lower. So you could conclude, if you believe in associations, that having type 1 diabetes is a predictor of survival. But I think what it really shows is that the people in the intensive treatment arm learned early that I have to pay attention to my diet and exercise and other things, but certainly not an excess mortality, if anything lower. Nevertheless, there's all the acute and chronic complications associated with diabetes. It's difficult to maintain good glycemia control. It's very expensive. 40% of the adults in the hospital today have diabetes. It's a major public health issue. So if we could come up with a cure, the patients and the economy would clearly benefit. So this is the Eisenbarth model that I say we've been studying now for 35 years or pursuing from 35 years. It's named after George Eisenbarth, who was a Duke colleague of Neil's and mine way back when, before he went to Joslin. And Dr. Eisenbarth posited that we're all born with a certain number of beta cells in our pancreas. But then if you are genetically susceptible to get type 1 diabetes and something happens, that an immune process ensues where the body's immune system kills the insulin-producing cells in the pancreas in a very slow process. Notice it takes years, all of which is silent to the patient until the patient loses a significant portion of their beta cell mass, at which time diabetes onset ensues. that process continues until they lose all their beta cells, and then they have brittle diabetes. That's the model. So whenever you see a model like that, I want you all, and you've heard me say it, say, well, what's the data for it? And the data is actually quite strong. That if you look at family members of an index case with type 1 diabetes and wish to know which of the family members are likely to go on to get the disease, you can measure anti-islet autoantibodies, and they will predict a high-risk group that with time can go on to get type 1 diabetes. I hasten to add that these antibodies are not thought to be pathogenic, but just markers of an ongoing immune process in the pancreas. If you look at the pancreas of someone who dies shortly after or even years after they've had a diabetes, you can see T-cell infiltrates in those islets called insulitis. There's a strong genetic component associated with risk for type 1 diabetes, best measured and best communicated by looking at the concordance rate among identical twins, such that if one member of an identical twin pair gets diabetes, the chance that the other one will get it is 50%, which already tells you that, that there must be an environmental component, or it would be 100% among identical twins. We know that various immune interventions at disease onset can prolong the honeymoon. And the last bit of data that I think most strongly ties the immune system to T1D pathogenesis along the lines of the Eisenbarth model is what I call Dr. David Sutherland's do-over experiment. Now, one of the themes I said I wanted to talk about was new versus old. I think we tend to think that insulitis is a newly observed phenomenon, and this is a slide that Sally Kent created and shared with me. But in fact, we've known about T-cell infiltration of the pancreatic islets for over 100 years, so it's really not so new. And then Willie Gepps and colleague Lecomte published it now 50 years ago. So it's been known for some time that islets in the pancreas of someone developing type 1 diabetes have a T-cell infiltrate. We believe that the killing process that destroys or at least eliminates the function of pancreatic beta cells in someone with type 1 diabetes is mediated by the T lymphocyte. APC in this instance stands for antigen presenting cell, but think of this as the beta cell. T cells don't recognize cells directly. Rather, a T cell receptor recognizes a human leukocyte antigen on that cell, and that human leukocyte antigen presents a small peptide of proteins found within that cell. And years ago, when I was in medical school, we thought that this recognition process was sufficient to activate this T cell. But then subsequent work demonstrated that, in fact, if a T cell recognizes its antigen presented by an HLA molecule and doesn't get a second signal, not only is that T cell not activated, it's permanently inactivated or anergized. And about 35 years ago, 40 years ago, Kevin Lafferty came up with the two-signal model of T cell activation, which was built upon a two-signal model of B cell activation. But that's what that model suggested was that in order for a T cell to become activated against an antigen presented by an antigen presenting cell, it not only needed this T cell receptor antigen driven signal, but also a second signal, or was called a costimulatory signal. And the first such costimulatory receptor pathway that was described was CD80, or then called B7, interacting with CD28 on the T cell. If signal 1 is generated through the recognition, and this is kind of like an on-off switch, then this T cell becomes activated and it will make new copies of itself and secrete cytokines and other T cell mediated products that will result in at least diminished function and probably killing of this cell. So I'm going to point out, you are all aware of the genetic risk factors for type 1 diabetes, and I'm just going to enumerate them because if you know this simple cartoon, you know 5 of the top 9 genetic risk factors for type 1 diabetes. Number 1 is the HLA molecules, and that's by far the most significant genetic association with type 1 diabetes pathogenesis. Number 2 is the antigen. We know that individuals with certain polymorphisms in the insulin gene are more likely to develop type 1 diabetes. So insulin is a known antigen attacked by the immune response in people with type 1 diabetes. 3, we know that there's genetic linkages with T cell receptor signaling pathway genes, and I'll show you those in a second. 4, when a T cell is activated, it starts making IL-2, and we know that there are genetic polymorphisms in the signaling pathway for the IL-2 receptor. And last, while there's no genetic risk of CD28 with type 1 diabetes susceptibility, another antigen that can come up on a T cell surface when it's activated is the gene called CTLA4, which shuts off the immune system. This one turns it on, and once the T cell's activated, this one comes up and turns it off. And people with polymorphisms in CTLA4 are at increased risk for type 1 diabetes. So many times, I know you've seen this slide published in New England Journal now 10 years ago of the various genes associated with susceptibility for type 1 diabetes. And as I've mentioned, if you just know that simple cartoon, you can account for 5 of the top 9. The HLA class 2, the insulin gene polymorphism. These are genes associated with signaling through the T cell receptor and the IL-2 receptor. And this is CTLA4, which shuts off the immune response. Now, having said that, to this day, we don't know if this huge risk associated with the HLA class 2 molecules is because those are particularly good at turning on an immune response against the beta cell, or particularly bad at shutting off an immune response against the beta cell. So that's one question, huge question, that remains open. This 2-signal model is still very much in front and center in immunology research. I show this figure only because it was published in New England Journal about 6 months ago by Jeff Bluestone and Mark Anderson. But on my simple cartoon, here you see T cell activation. Here's the T cell. Here's, let's say, the beta cell. This is the T cell receptor recognizing a peptide presented by the MHC. And here's CD28 interacting with the CD80 or CD86 to turn on an immune response. And then once the immune response is turned on to inactivate it, CTLA4 expression is upregulated, and it turns out the affinity of CTLA4 for these CD80 and CD86 is greater than CD28, and so the immune response is dampened down. Also in that review, however, this T cell co-stimulation field has exploded over the last 30 years. And I draw your attention in particular to the PD-1, which is expressed on T cells, and PD-L1, which can be expressed on antigen-presenting cells like beta cells. And I wish for you all to recall that a few weeks ago, Sally Kent presented that when we isolate, she isolates T cells from the islets of someone with type 1 diabetes, she finds a population of these PD-1 expressing T cells, implying that the immune response is trying to dampen down or to be inactivated against the human pancreatic beta cell. And last, I like to cite this as the strongest bit of data showing T1D as the immune-mediated process. David Sutherland years ago did an identical twin partial pancreas transplant for twins that were discordant for diabetes. In other words, this twin, and in each case it was women, so I'm showing women here. One member of an identical twin pair was diagnosed with diabetes as a child, And if 15 years went by and the other sister didn't get diabetes and didn't have anti-islet antibodies and had normal glucose tolerance, with consent, Dr. Sutherland took the non-diabetic sister to the operating room, removed half of her pancreas, and transplanted into the diabetic sister, thinking that he could cure this diabetic sister. And what happened was, in every case, the diabetic sister, within a few months, destroyed the beta cells that had been transplanted a few months earlier. And so the immune system of this sister, even though in one instance it had been 29 years since her diagnosis and the transplant, quickly destroyed those beta cells again. It's as though the immune system learned, I thought I told you 29 years ago, I don't like beta cells. If you give me beta cells, I'm going to kill them again. And it did. But this has always intrigued me, and there's lots of reasons why it might be. Why does it take, when a person with diabetes is developing the disease, it takes months or years or even decades to kill the small number of beta cells in the pancreas? And yet when Dr. Sutherland sews in a new pancreas from an identical twin sister, the process takes place over a few weeks. It could be the surgical trauma and taking the pancreas out and putting it in ice for a little while, I don't know. But it's intrigued me that this immune response is very rapid, where the native one is very slow. So now I'm going to jump into some of the things that we've always known about type 1 diabetes, and which I hope to raise questions in your mind today about. And I say this is an outline of my talk by colloquialisms. Churchill said, men occasionally stumble over the truth, but most of them pick themselves up and hurry off as though nothing has happened. And I'll show you plenty of examples where we've shown over and over again that something isn't true, but we still say it. Mark Twain said, it's not what you don't know that kills you, it's what you know for sure that just ain't true. George Box admonished us to remember that all models are wrong. The practical question is, how wrong do they have to be to not be useful? And last, the great Sir William Osler encouraged us all to have a stern iconoclastic spirit, not passive skepticism born of despair, but an active skepticism born of a knowledge that recognizes its limitations and knows full well that only in this attitude of mind can true progress be made. So this is another slide that Des Schatz shared with you a few months ago and allowed me to show. We've done billions of dollars of interventional studies looking at antigen-specific and non-antigen-specific immune-modifying therapy for those with type 1 diabetes over the last several decades, and none of them have led to a change in the way that we manage someone with type 1 diabetes. So you have to ask yourself, what's the problem? We've got the model. It's an immune-mediated process. We built a strong case for this being an immune-mediated process. Why haven't these therapies worked in people? And at least one possibility is that the rodent model that we've used to study the immune process underlying type 1 diabetes may have steered us wrong. Or it could be that the beta cell is actually not just a passive victim in this immune response. Maybe there's some problems with the beta cells themselves. Maybe human disease is much more heterogeneous than we've thought, and so that if we could subcategorize people and know what specific therapy to give them, that we may succeed. Perhaps the timing of our treatment matters. I showed you that type 1 diabetes, or I've alluded to the fact that type 1 diabetes takes many months and years to develop. Maybe the timing of the immune treatment is important. And maybe we just don't understand the autoimmune process very well. My former boss at the NIH, Alan Spiegel, who then went on to be the dean at Einstein, used to testify before Congress and would tell them all, they would say, you know, Dr. Spiegel, we've invested billions of dollars. Why haven't you cured this disease? And he would always say, Senator, this isn't rocket science. It's a lot harder than rocket science because we don't understand the process yet well enough to specifically intervene. So this is another slide that Shat shared. These are all the therapies that work in the non-obese diabetic autoimmune diabetes mouse model. And they all are effective, and none of them have worked in people. So at some point you have to say that I think there's 507 trials on here. You'd think at some point someone would say, gosh, maybe we shouldn't use the NOD mouse model as a way to predict what might work in people, because 507 out of 507 times, those successful results have failed in people. And yet we pick ourselves up and carry on as though nothing has happened, or we have for decades. And what is particularly concerning to me is that there are other therapies that we might have tried in people, but we didn't because they didn't work in the NOD mouse. So we were following NOD mouse as an arbiter of true north, and at least this data would suggest that it may not be. I like to show this slide because it's old, 80 years old, 90 years old. But the assumption that we all have the same amount of islet or beta cells in our pancreas has not been known to be wrong for decades. This is an old study by Ogilvie where he used pretty non-sophisticated techniques to know the amount of islet weight in different humans. And as you can see, you don't have many islets as a newborn. They grow until there's a peak of growth until like 10 years of age, and then another one through puberty. But then the number of islets stays pretty stable. That could be because as you lose beta cells, your body keeps pace. Or it could be that we develop a certain number of beta cells by age 25, and then you live with those beta cells for the rest of your life. And I'll show you some data that suggests the truth is the latter. Well, that study by Ogilvie, which was 37, was rediscovered by Dr. Butler and colleagues and published in Diabetes Care in 2013, where they looked at the number of beta cells in non-diabetic individuals. And they not only found a correlation with age, I don't show that here, but our assumption that we all have a certain number of beta cells in our pancreas is just wrong. These are non-diabetic individuals, and note that there is a 5 to 10-fold variability in the number of islets or beta cells in the pancreas of these individuals. This isn't the only study that has found that. There's other studies that find the same thing. So if you look at, for instance, a human liver or a human heart, they're pretty much the same size from person to person, not huge variability, maybe 20, 30% difference in weight if the liver's not fatty infiltrated. But in the case of human beta cells, there's a 5 to 10-fold variability in the number of beta cells, and we don't understand why. Not only that, we know that individuals who go on to get type 1 diabetes have smaller pancreases. And this has been a subject that's been developing over the last 5 years. Jason Gallia, for instance, published that if you look at the pancreas of someone with recent onset type 1 diabetes by MRI, or measuring the volume of the pancreas by imaging studies, that if you have recent onset diabetes, compared to non-diabetic individuals, the pancreas is about 40 to 50% smaller. And remember that islets only comprise about 2% of the mass of the pancreas. So why would the whole pancreas be smaller if it's a beta cell specific disease? This more recent paper by Martha Campbell Thompson and colleagues at University of Florida took that one step further. And because family members of an index case with type 1 diabetes are at increased risk for developing diabetes, they looked at first-degree family members, those that either had multiple autoantibodies or single autoantibodies, or even those that didn't have autoantibodies. And they found statistically significant smaller pancreases in families of an index case of type 1 diabetes. So the entire pancreas of someone in a family with type 1 diabetes tends to be smaller than that in the general population. So the factors that regulate pancreas mass and beta cell mass are completely unknown. All right, now I'm going to turn to the famous DCCT, and you'll see why in a minute, I hope. But I've often told you that textbooks, when Marjorie and Neil and Mike and Leslie and I were in medical school, Samir, all referred to the glucose hypothesis. And I found one of my old pathology books, and I copied this out of it. It said atherosclerotic events such as myocardial infarction, cerebral accidents, gangrene, etc., are known to accompany type 1 diabetes. And the Joslin Clinic points with pride to the victory medal diabetics. But then notice it says most diabetologists doubt that metabolic control so directly influences the vascular disease, but nonetheless attempt to maintain reasonable metabolic control in the hope that you might prevent these vascular complications. It seems almost patronizing. It's kind of, isn't it cute that these diabetes docs try to control the blood sugar when it's really not related to the complications of the disease? Until this study came out, the DCCT, it enrolled 1,441 patients with type 1 diabetes, some of whom already had retinopathy, some who didn't, and half were assigned to receive intensive control and half conventional control. Now I want to dig into the details of the DCCT because it's very important that everybody know exactly how that was done. It enrolled patients between the ages of 13 and 39 years of age. Primary prevention and secondary prevention was purely differentiated by whether or not the ophthalmologist could see evidence of microaneurysms in the eyes. You could enroll in the study if you'd had type 1 diabetes diagnosed at least 1 year earlier and up to 15 years later. There was no retinopathy in the primary prevention group and minimal to moderate in the secondary intervention group. And the primary intervention group had no microalbuminuria, whereas the secondary intervention group could have some minimal albuminuria. So the one question I want you to ask yourself is, why did it take 50, 60 years to test the glucose hypothesis? And the answer was, as those of you of my generation know, we didn't have the tools to control glycemia control back in the 70s and 80s. We didn't start measuring patients' blood glucose to regulate insulin doses until the early 1980s. Patients were managed with what was called a double voided urine specimen to know, and the whole goal was to try to keep sugar out of the urine. So what did the study show? The conventional treatment arm was treated the way most patients were treated with diabetes up until 1993. That is, try to avoid the symptoms of hyper and hypoglycemia, give the patient 2 shots of insulin a day or less, have them monitor their urine daily, check a quarterly A1C, and see them quarterly. What the DCCT did was they tried to maintain near normal glycemia, keep the fasting sugar between 70 and 120, postprandial less than 180, and at 3 a.m. greater than 65. They sought to have an A1C of 6.05%. They advocated testing the blood sugar 4 times daily, and they had monthly visits. I think you all know that the intensively treated group succeeded. For 10 years, they maintained hemoglobin A1C average of around 7.5%, whereas the conventional treated group achieved what everybody achieved back then, an A1C of 9%. And then the study ended and found the profoundly beneficial effects on complications. But it would be even more profound. What most people don't realize is that even within the intensively treated arm, there were quite a few people with A1Cs of 8, 8.5, 9, even up to 12%. So these results, where you see this not only profoundly statistically significant improvement in eye disease, kidney disease, and nerve damage, but also clinically relevant decreases in those, would be even more profound if you just looked at people who had an A1C of less than 7%. So these majorly important decreases and statistically significant decreases are minimized by the fact that quite a few people really didn't do well. And then if you look at cardiovascular endpoints, MACE, heart attacks, strokes, sudden death, it took years later, but for enough events to accrue. But tight glycemia control also prevents those things. So this firmly established, the world changed after this study was published. Now it was imperative to try to improve glycemia control for those with diabetes. And I say another lesson that's frequently forgotten is that unfortunately, when these people that were enrolled in the DCCT were referred back to our general healthcare system, their control deteriorated, which I think is a damning evidence for the efficacy of our current healthcare system. And there's another couple fine points that I want to emphasize from the DCCT in the lessons forgotten category. I've already told you that there was a secondary prevention and a primary prevention arm. If you looked at the 855 patients in the DCCT who had had diabetes for less than 5 years when the study began, subsequent analysis showed that about 30% of them still made significant amounts of C-peptide, which they subsequently called responders. And another 2/3 did not have C-peptide. So years later, in this study that Jerry Palmer led, first thing they asked was, does it make any difference? So what if some of the patients had small amounts of C-peptide secretion? Did they do any different than the non-responders? And the answer is, were they any different? Yes, they were different. They tended to be older when the diabetes was diagnosed and had a shorter diabetes duration. But they also had a lower insulin requirement and better A1Cs and less background retinopathy. So having the pancreas make some insulin is important for how the patient does. The next question they ask is, we know from the DCCT that it's important to maintain good glycemia control. Now let's ask the question, if you're treated intensively and your pancreas still makes insulin versus doesn't, does this amount of insulin production help in the intensive management of their diabetes? And the answer is yes, it does. That these with significant C-peptide production from their pancreas had less retinopathy and fewer hypoglycemia episodes. And then the last question is, if you entered the study making C-peptide and you were treated intensively versus conventionally, did this make a difference in how much insulin your pancreas made? And the answer is yes, it does. That intensive control prolongs the pancreas's ability to make significant amounts of C-peptide. So the ultimate conclusion of the DCCT is tight control begets tight control, because controlling the sugars early allows the pancreas to make more of its own insulin, and any insulin made in the pancreas lowers the retinopathy risk, lowers the insulin doses, and lowers the risk for hypoglycemia. So important lessons that I think are forgotten about the DCCT. So since beta cells are surviving with good glycemia control, what is the mechanism of that? How is it that beta cells live longer in an intensively treated patient? Now, another question is, the Eisenbarth model said everyone goes on to lose all of their pancreatic insulin producing capacity eventually when they have type 1 diabetes. And again, we've known for decades that that just ain't so, but there have been several studies that have looked at that, and I'm going to cite several of recent ones for you. The most sensitive way to look for pancreatic insulin production is with the urinary C-peptide to creatinine ratio. And this is a large study done by Richard Oram et al. From Exeter, England. And what you see is if you measure the amount of urinary C-peptide versus creatinine, most people with type 1 diabetes still have detectable amounts of insulin production from their pancreas decades after disease onset. And this is the level, 0.6 nanograms per mil, where it's clinically very significant. This has also been found in the type 1 diabetes exchange data done in the States and a bit in Canada, where if you look at individuals diagnosed with type 1 diabetes after the age of 18, again, 40 years after disease diagnosis, 16% of them have significant C-peptide production from their pancreas. So what? I've already told you that C-peptide production from the pancreas makes it easier for that individual to control their diabetes. And I point out to you all what are called the adjunct 1 and 2 studies. They looked at adding Victoza or liraglutide to a patient with type 1 diabetes. There were 2 studies. One, adjunct 1, when the goal of insulin treatment was to treat to target. Adjunct 2 just gave a fixed basal dose of insulin and added liraglutide. These were, and the endpoints were glycemia control, daily insulin dose, and weight. You can read the inclusion criteria. You had to be diagnosed with type 1 diabetes at least a year earlier, have a baseline A1C between 7 and 10%, and be on a stable insulin dose for 3 months or more. The patients in these studies had diabetes for about 20 years on average, and the mean baseline A1C, just like in the follow-up of the DCCT and in the rest of the country, is about 8% baseline mean A1C. So patients were either treated with their usual insulin doses and started on liraglutide or placebo, and what did they find? Well, for adjunct 1 and 2, I don't expect you to see the fine details on this, but in all cases, the A1C was better, the insulin dose was less, and patients lost weight with increasing doses of a GLP-1 receptor agonist, whether it was treat to target or fixed basal insulin dose. However, you all don't know this because it didn't reach statistically significant difference the way they defined the study. But what I want to point out to you is the subgroup benefits for those with detectable C-peptide. And here we're looking at a fairly consistent theme is that about 15% of people, even 20 years after diagnosis, still make significant C-peptide. And what you can see is that those who got liraglutide or a GLP-1 receptor agonist had statistically significantly improved glycemia control compared to those who did not get the GLP-1 receptor agonist. Now, this is a subgroup analysis, and you're not supposed to go back and reanalyze data when it's done, but the number of patients in these studies was pretty significant, and I contend this is good data to show that a GLP-1 receptor agonist is useful in someone with type 1 diabetes, especially if they're overweight, and especially if you can demonstrate a C-peptide level, but measure a C-peptide in their serum. So this is the urinary creatinine ratio that I already showed you. Just another figure from that same paper showed that if you look at any measurable pancreatic C-peptide, no matter the duration of diabetes, you can find pancreatic insulin production in the majority. It's not a lot, but it's there. So one question is, how do some beta cells survive the ongoing anti-beta cell immune response? And that's an open question. Those types of observations, however, have led to reviews like this, written by Richard Oram, Emily Sims, and Carmela Evans-Molina, that was in Diabetologia a couple of years ago. And they wondered if maybe we're constantly making new beta cells, and I'll show you some data about that. Maybe there's an altered immune response after you've had diabetes for a while, or maybe there's something different about the beta cells that survive in the pancreas. So we'll spend the next couple slides talking about the capacity for human pancreatic beta cells to proliferate or regenerate. I want you all to recall Sally Kent's talk from a few weeks ago when she has identified CD57, which is a marker of T cell exhaustion, and PD-1 positive CD8 T cells in individuals with type 1 diabetes of 10 years duration. And if you think back to what I said about the costimulatory pathway, PD-1 is a signaling receptor that downregulates T cells. In other words, tries to put the brakes on them. And then we and others are trying to explore what's unique about the beta cells that survive in a human pancreas despite decades of type 1 diabetes. So let's talk about beta cell turnover. What's the capacity for human beta cells to regenerate over time? And what you're looking at here is what's called the bomb curve. Nuclear physicists have known for 60 years precisely how much C14 is in our atmosphere. And it turns out that in the 1950s and early 60s, before Linus Pauling put an end to it, there was nuclear atmospheric testing of nuclear bombs, so that the amount of C14 in our atmosphere rose precipitously until that was stopped. And it's been gradually decreasing since. That's not because the C14 is decaying, because the half-life of C14 is nearly 6,000 years. It's decaying because all of the C14 in our atmosphere is dissolving in the oceans and being made into trees and all organic materials. So recognizing that, Dr. Christy Spalding in Sweden, now 15, 20 years ago, isolated the DNA from tree rings. And she measured the amount of C14 in the tree ring DNA. Since she knew exactly when that DNA was made, she thought maybe the amount of C14 in the DNA correlates with when the cell was made. And in fact, she found that very tight correlation. That if DNA was made in 1963, there was a lot of C14 in that DNA. And then since then, it's been declining. So we decided to look at the amount of C14 in brain-dead donor pancreatic beta cells. And these are very hard studies to do because you need a lot of beta cells, and there's not a lot of beta cells in the human pancreas. So we tried many times, but we were able to isolate sufficient numbers of beta cells in 2 different donors when this study was published. And it's being replicated now by Yuval Dor in Israel. But in any case, the first was a woman who died of head trauma at age 80. She was born in 1926. And when we studied the amount of C14 in her beta cells, the data suggested that her beta cells stopped making new copies. There were no new beta cells in her pancreas after about 1955. So that she lived until 2008, I think. And for the last 50 years of her life, she was living off of beta cells that were made when she was 30. And we got the same conclusion from a second donor who was born in 1958. So using this C14 DNA content data, we argued that you can make new human beta cells until you get to age about 25 or 30, which is about what Ogilvie saw in 1937. And then you live with those beta cells for the rest of your life. Well, it's always good to have another technique to look at beta cell turnover. And so Shira Pearl, who was a fellow at the time at the NIH with me, discovered that the NIH had in its pathology vault pancreases of people who had been treated with thymidine analogs years earlier as part of a clinical trial for cancer chemotherapy. Not only the BRDU and IDU were thought to be radiosensitizing agents, but also the patients were treated with these to measure how quickly their tumors were turning over. In any case, she found patients who had been treated with BRDU or IDU prior to their death, and when they died, their pancreas was still in the vaults at the NIH Clinical Center. So we went back, pulled out those pancreas sections and stained the pancreas sections to see if their beta cells stained with IDU or BRDU. And what you see here is the kind of cancer that the individual had, their age when they were treated with the IDU, their weight, and then how long after they got the thymidine analog they survived. And what you see is that if the 2 individuals that were young, 18 to 20, 1 to 2% of the beta cells in their pancreas were stained with IDU or BRDU, indicating that their beta cells were turning over. But then if we looked at the other 10 patients who were older when they got their cancer, we never saw a single beta cell stained with IDU or BRDU, indicating that beta cells divide until you get to age 25 or so, and then they stop. So I don't think there's much capacity for human beta cells to proliferate after age 25 or so. Certainly we saw no evidence of it. Okay, so are beta cells being killed by the immune response or just inactivated? This is, several of us just attended an NPOD meeting, virtual NPOD meeting about a month ago, and this is a very hot topic. Emily Sims and Richard Oram and others have been measuring the amount of proinsulin in the bloodstream of someone diagnosed with type 1 diabetes. And they also measure the amount of C-peptide in the serum. And as I've mentioned, if you have diabetes for less than 10 years, you're pretty likely to still have measurable C-peptide levels. And if you measure super sensitive C-peptide levels, it's only a small number that have lower than detectable limits C-peptide. The surprising thing is that if you measure the amount of proinsulin, I didn't mean for that to disappear. The proinsulin level, even in those that don't make C-peptide, is still about the same as those that do make C-peptide. And this is data that's used to argue that beta cells haven't died, they've just de-differentiated, or they've just lost their ability to secrete insulin, which is an interesting topic. There's other people, Peter Arpin at University of Michigan, stains pancreatic sections for proinsulin and finds a lot of apparently insulin negative cells that will immunostain for proinsulin. We've all been taught that type 1 diabetes is a beta cell specific disorder. Is that true? Well, here's a 30-year-old study that says maybe not, because if you look at the amount of fecal chymotrypsin, which is an acinar exocrine pancreas product, those with type 1 diabetes have significantly less fecal chymotrypsin than controls. And similarly, 20 years ago, another group studied the amount of fecal elastase, another exocrine pancreas product. And again, those with type 1 diabetes have statistically significantly less fecal elastase than those with type 2 diabetes. So taken together with the pancreas size data that I've shown you earlier, and now this functional data, it's often stated that type 1 diabetes is a beta cell specific disease, but that's not consistent with either anatomical or functional data. So the question is, are there pancreatic abnormalities observed in type 1 diabetes a cause or a consequence of the beta cell failure? Here's another thing that you'll hear all the time. We hear that you can predict who's going to get type 1 diabetes now with great reliability. If you look at first-degree family members of someone with type 1 diabetes and measure the proportion that develop diabetes over time, it's true that 8 years later, you've identified 70% of those who are going to get diabetes, but 30% still don't have diabetes. And notice that it's a very slow decrement to move toward disease pathogenesis. We know that type 1 diabetes has a very long prodrome. One of the studies that the TrialNet group does is called a natural history study. If you are at high risk for diabetes and you have antibodies, they follow you with every 6-month mixed meal tolerance tests. And they reported a few years ago that 80 patients never seemed to progress to type 1 diabetes, where another 36 did. And then if they looked back in time at the amount of C-peptide that person would make, the progressors would make, you can see that it was gradually decreasing over a 3-year, 2.5-year period, suggesting that this immune response is very slow. And I've mentioned the identical twin data, other data that George Eisenbarth published now. He started an identical twin study 30 years ago. And what most people will focus on is that if one member of an identical twin pair gets it, the other one will get it pretty quickly thereafter. But in half the identical twin pairs, let's look at this one. This identical twin developed diabetes at age 8, and his brother developed it at age 15. Or some other extreme examples, this one developed type 1 diabetes at, let's say, 12, and the identical twin developed it at age 45. So even though they're genetically identical, raised in the same environment, there can be a huge disparity in when identical twins get diabetes, and we don't understand that disparity at all. Sally Kent presented this data a few weeks ago, but I thought it was worthwhile bringing up again. Everyone knows that type 1 diabetes is a childhood onset disease, except that it's not. This is a study that looked at 61 million commercially insured Americans from 2001 to 2015. They called it type 1 diabetes. If the billing material said type 1 diabetes, if they were filling insulin prescriptions, and 182 days of insurance coverage had existed. They excluded people with gestational diabetes, and you could only be enrolled if you were less than age 65. But as you can see, if you measured the area under the curve of these two figures, you are actually more likely to be diagnosed with type 1 diabetes after age 18 than before it. So you could say it's an adult onset disease, just with peak incidence at around age 15. We know that loss of islet function is not linear over time. This is the amount of C-peptide made by individuals diagnosed with type 1 diabetes at various time points after their disease is diagnosed. And you can see that for the first year after disease diagnosis, C-peptide loss is at a certain pace, and then thereafter it slows, perhaps because PD-1, CD8 T cells come up, as Sally presented a few weeks ago. But we don't understand this different kinetics of the disease. And in clinical trials, we're just oblivious to that. When someone comes in, if they've been diagnosed within a certain period of time, we give them the immunotherapy, not recognizing that different people may have different disease kinetics happening at that time, or based upon their age, which is shown on this slide. This is just looking at individuals in various trial net studies of different age range, at how quickly they lose their C-peptide after disease diagnosis. And for those who were diagnosed with diabetes below age 12, they lose it at a fairly rapid pace, whereas the majority that develop diabetes after age 18 have persistent C-peptide production for quite some time. And as I've already shown you, that can be influenced by the level of glycemia control achieved. So last, what triggers this anti-beta cell immune response? No one knows. We should ask Jennifer Wang to present her work on a potential viral pathogenesis, and she's doing some very good work here. And once triggered, does it ever self-regulate? There's a well-funded, well-studied group called the Environmental Determinants of Diabetes in the Young, or the TEDdy study. And they looked at individuals with a single autoantibody who went on to develop multiple autoantibodies or reverted to antibody negative. These were in high-risk HLA types. And you can see once you developed an autoantibody, the chance that you'll develop multiple autoantibodies is about 75%. And these are the individuals at highest risk for sometime over the next 8 to 10 years developing diabetes. But another surprise is that about 23% of people become antibody negative after they are antibody positive. So the autoimmune process may start and be regulated down to off in a significant number of people. If we treat these people with immunosuppression because they have autoantibodies, they might never have developed disease, probably wouldn't have. So this is just the proportion without type 1 diabetes in the TEDdy study. As I said, if you have multiple autoantibodies, which is this line, you're about 70% likely to get diabetes over the next 7 years. But if you are autoantibody single autoantibody positive or you revert to negative, you don't get diabetes. I'm just going to conclude with a few slides explaining what we're doing to try to explore diabetes pathogenesis. This is a team effort between us and WPI. When I say us, Sam Reddick, Dave Blodgett, me, Sally Kent. We are getting viable human pancreatic slices from the NPOD group of individuals who have died either with or without diabetes. And I'm showing a magnified version here, but this is about a centimeter by centimeter square, and it's only a few hundred microns thick. We can look at that under a microscope and identify, as we've shown here in red circles, what looks to us like islets, and then we can stain. And in fact, what we thought were islets were islets. The staining turns the islets red. We want to know if the cellular constituents of an islet influence that islet's function. We want to know if when T cells infiltrate the islet, that influences the alpha-beta cell ratio, or if that influences beta cell function. So what we do is that we take a Petri dish and put the pancreatic slice in it, and with our WPI colleagues, and Sam Reddick is driving this mostly with them, we've come up with a system where we can punch out individual islets and put them in individual wells and then characterize the cells within that islet and that islet's function so that we can begin to answer these questions. And remember, the DCCT couldn't be done for 50 years after insulin was discovered because we didn't have the techniques. We're putting a lot of effort into the techniques to study individual islets from the pancreas of individuals with type 1 diabetes. And then last, what we call our Manhattan Project, which we do collaboratively with the Jackson Labs and Harvard Stem Cell Institute and other colleagues at Harvard. We are now taking peripheral blood cells from donors, diabetic or not. Dr. Melton's lab can turn those into stem cells. He can then turn those into what he calls SC beta cells. Renee Marr in our group can turn them into thymic epithelial cells. And George Daly is working to develop them into hematopoietic stem cells so that we can put these types of cells into a mouse with no immune system, destroy this mouse's beta cells so that it's dependent on the human beta cells for survival, and then watch the disease process happen in these mice over time. That's a huge collaborative effort led by Doug Melton with us as partners, funded by the NIH Human Islet Research Network. And then just a few weeks ago, the same general team members were recognized as the third Center of Excellence by the JDRF, that's called the New England Center of Excellence, to see if we can create stem cell-derived beta cells that will evade the immune-mediated destruction underlying type 1 diabetes. Now, I have to say, I don't want to be hypocritical because this is a beta cell-specific focus still, and I've already shared with you that individuals with type 1 diabetes also have general pancreatic defects that we don't understand.